Welcome back to the deep dive. If you've ever had jet lag, pulled an all-nighter, or, just felt that crazy 3 p.m. slump, you know your body is a deeply rhythmic machine.
It absolutely is.
Today we are diving deep into the conductor of that whole symphony, the circadian rhythm, and how it really truly orchestrates the entire brain.
And this rhythm is, it's more fundamental than most of us realize. It's not just about when you feel tired or awake.
Right.
These cycles are the essential temporal regulators of basically everything from your sleep and wakefulness to hormone secretion, metabolism, even how sharp your cognitive function is.
And here's the hook, the real problem. When the internal timing gets thrown off, whether it's from shift work or just genetics, the consequences can be, well, they're severe.
Yeah, we're talking about an increased risk for serious metabolic disorders, cardiovascular dysfunction, really complex neurological conditions.
The brain has to keep time perfectly, but for decades, we've only had these fragmented clues about how much of the brain is actually in on the timekeeping.
And that's the core of the knowledge gap we're talking about today. I mean, we've all focused, and rightly so, on the master pacemaker, the suprachiasmatic nucleus, or SCN.
Deep in the hypothalamus.
Exactly. We know the SCN is incredibly complex. It's made up of all these different neuronal populations like EVP, VIP, and GRP-expressing cells that are all sort of chattering with each other to set the main clock.
But trying to map that rhythm in the rest of the brain has just been messy. So inconsistent.
It really has. Previous methods, think traditional slicing, electrophysiological recordings, sparse sampling, they were essentially like looking at individual stars without ever seeing the constellation.
You're just getting tiny little dots.
Tiny dots. They lacked the spatial continuity to see if the whole brain was really ticking along on its own schedule.
Okay, so that brings us to the breakthrough at the heart of this deep dive.
Precisely. Researchers found a way to finally bridge that gap. They used this innovative high-throughput 3D imaging tech to create a single-cell atlas of neural activity.
And not just in one brain.
No, across 144 whole mouse brains, all collected in a precise time series. So the mission for you, the listener, is to really get how they did this and what it means when we find out just how synchronized the brain truly is.
So let's unpack that, because the headline is just staggering, that nearly 80% of the entire brain is rhythmic. But to believe it, we need the how. The methods here are the real story.
It all starts with the conditions. They took adult male mice and sampled their brains every 4 hours over 2 days.
So it's 12 time points.
12 time points, 144 samples in total. And crucially, this entire experiment was done under constant darkness.
Constant darkness. Why is that so essential? I would think a normal light-dark cycle would make the rhythm easier to spot.
But that's the catch. If you use a regular light-dark cycle, the environment is just telling the brain what to do. It entrains the activity.
It's an external cue.
It's a huge external cue.
Right.
By removing all light and keeping them in constant darkness, you force the brain to rely purely on its own self-sustained internal clock. It lets you measure the true autonomous rhythm.
Okay, that makes sense. So now the technique that lets them see the whole picture, CUBIC. It sounds like something out of science fiction. How do you make a brain transparent?
CUBIC stands for Clear, Unobstructed Brain Imaging Cocktails and Computational Analysis. And it really is a chemical process that fundamentally changes the tissue.
How?
Well, it works by adjusting the refractive index of the tissue and pulling out lipids, which are what make it opaque. The end result is that the entire intact brain becomes transparent.
You're kidding. Transforming a foggy pane of glass into a perfectly clear crystal.
That's a great analogy, exactly. And the incredible benefit here is that you preserve the entire 3D structure. This solves that spatial continuity problem we talked about. You can see every neuron in its proper context.
So once the brain is clear, you still need to know which neurons were actually active at each time point. For that, they use c-Fos.
Yes, c-Fos. It's an immediate early gene, an IEG. You should think of c-Fos as a tiny biological receipt.
A receipt. I like that.
Yeah. So when a neuron fires up, gets a burst of activity, it triggers the rapid transcription of this c-Fos protein. So the protein acts as a really reliable indirect marker that says, hey, this cell was active in the last couple of hours.
Okay, so they clear the brains, they stain them for these c-Fos receipts, they image the whole thing with light sheet microscopy, and then they have to make sense of this mountain of data.
And that analytical pipeline is key. They used a supervised machine learning algorithm to automatically detect every single c-Fos positive cell and we're talking millions, and then map their exact 3D locations onto a standardized brain atlas.
So you can compare apples to apples across all 144 brains.
Precisely. But before you can claim anything, you have to check your work against the gold standard, the known clock.
The SCN.
The SCN. And it performed exactly as you'd hope. It showed a clear 24-hour rhythm peaking during the subjective day around circadian time 5.3.
So the pipeline works.
The pipeline was validated top to bottom.
So the conductor is keeping perfect time. But when they looked beyond the SCN, what was the seismic shift here?
The finding is that the rhythmicity it's just everywhere. Of the 642 brain regions they annotated, 508 of them showed significant circadian rhythmicity.
508. Wait,
that is a stunning 79% of the brain operating under a consistent internal 24-hour rhythm. I mean, it just fundamentally changes how we should think about timekeeping in the brain.
Okay, so let's pause for a quick recap of how we got to this incredible number.
Right. So first, the big challenge was that our previous maps of circadian rhythm were fragmented, kind of like looking at a city through a keyhole.
And the solution was this amazing new method, CU by C tissue clearing, which makes the whole brain transparent, combined with mapping and activity marker, c-Fos.
And they did this on 144 mouse brains, all kept in constant darkness to isolate the brain's internal clock.
Which led to the headline result. Nearly 80%, or 508 out of 642 brain regions, are significantly rhythmic. The clock is brain-wide.
When we start looking at the actual timing, the story gets even more interesting.
Right, because if you just look at the raw data, the sheer number of active cells, there's a huge peak of activity during the late subjective night.
CT20 to 24, yeah.
Yeah.
Which makes sense for nocturnal mice, right? That's their active phase. But the magic happens when you start organizing these regions by function. That's where you see this story of precise temporal specialization.
Okay, let's break down those functional groups. It's almost like the brain has its own internal daily schedule. What about something basic, like the wake-sleep seesaw?
The opposition is just perfect. You have these regions that secrete powerful wake-promoting neurotransmitters.
Like orexin, serotonin.
Exactly. The lateral hypothalamic area for orexin, the dorsal raphe for serotonin, the tuberomammillary nucleus for histamine. They all peaked late in the subjective night between CT20 and 23.
That timing lines up perfectly with what's called the wake maintenance zone. So they're not just kicking things off at dusk, they're firing to sustain peak wakefulness right until the end of the active period.
Precisely. Now contrast that with the sleep-promoting centers. The ventrolateral preoptic area, the VLPO, which actively shuts down those wake centers.
It peaked at the exact opposite time.
The exact opposite. Mid-subjective day, around CT 6.5, which is the mouse's deepest inactive phase.
Yeah.
Wake peaks late night, sleep peaks midday. A perfect internally driven antiphase rhythm.
That's beautiful coordination. But where it gets really, really interesting for me is in cognition. The memory system showed a functional split that seems incredibly strategic.
It's fascinating. The hippocampal formation, which is the hub for memory, is split by time. So the regions associated with memory retrieval and integrating information, specifically CA1 and CA3.
They peaked late at night.
Right alongside the active phase, CT21 to 22. Makes sense. If you're out exploring the world, you need your retrieval power to be at its peak.
So your ability to use what you already know is maximized when you're active. But what about making new memories, the encoding part?
Those systems peaked at the opposite time. The dentate gyrus, which is crucial for what's called pattern separation, encoding new, distinct memories, it peaked mid-subjective day, CT 6.9.
So when the mouse is quiet and resting...
Exactly. When it's not being bombarded with new stimuli, the brain seems to be prioritizing encoding new information during the quiet, inactive phase. It separates memory storage from memory retrieval in time.
Wow, it's like the brain has these two different modes. A daytime operation for filing new memories, and a nighttime operation for actively using them.
We see that same kind of anticipation in the sensory systems, too. Visual-related areas, like the superior colliculus and parts of the visual cortex, they all peaked during the subjective day.
Even though they're in total darkness.
Right. This isn't a reaction to light. It's the brain internally preparing, like an internal weather forecast for the day ahead.
And the opposite for hearing.
And the opposite for hearing. Auditory regions, like the inferior colliculus, peaked during the mid-subjective night. The system is optimizing vision for its subjective day and hearing for its subjective night, all without a single external cue.
This region-level data is already so powerful, but timing in the brain can be more nuanced than that. If you zoom in with this technique, Do you see gradients within a single region? Does the whole SCN just flash on at once?
No, not at all. And that's where the high-resolution voxel-wise analysis comes in. We're talking a 20-micrometer scale.
Yeah.
And what you find is this immense internal heterogeneity. The timing is encoded down to a tiny sub-regional level.
Okay, so give us an example. The master clock itself, the SCN, how does it break down?
So the SCN, which looked like one coherent unit before, actually shows this gradual internal wave of peak times. The anterior dorsomedial part peaks earliest, around CT2 to 4, and then it washes over to the ventrolateral area, which peaks later from CT4 to 8.
So it's not one big flash, it's a controlled wave.
A controlled wave. It reflects the known cellular organization, the core and shell regions, working together to make the clock robust.
So even the conductor has different sections playing their parts at slightly different moments. Did you see this in the memory systems too?
They did. In CA1, they saw a gradual peak time shift along its dorsoventral axis. And remember, the entorhinal cortex, the medial part, which does spatial stuff, peaked during the light phase, CT8. But the lateral part, for non-spatial cues, peaked late subjective night, CT20-24.
The function is literally reflected in the internal timing of the structure.
It's all there in the timing.
Okay, but there's a problem, right? You said most of the brain's raw activity clustered late at night. So how did the researchers untangle the full 24-hour cycle of coordination from that one big peak?
They needed to shift their perspective, and this led to a really ingenious analytical move called the cell count ratio.
Cell count ratio, what is that?
Instead of just counting the active cells in a region, they divided that number by the total number of active cells in the entire brain at that same time point.
Ah, okay. So instead of asking, is this region active right now, you're asking, when is this region most active compared to everybody else?
Exactly. And this normalization revealed a completely different picture. The relative peak times were much more evenly spread across the entire 24-hour cycle. And this structured diversity is what leads to the most important result of all.
Which is?
The prediction of internal time.
They can look at one brain at one moment and tell you what internal time it is.
Yes. That broad structured distribution of relative peaks means a single snapshot of the brain's global activity can be used to accurately infer the organism's brain circadian time.
How? What did they use?
They adapted these really clever computational methods that were first developed for gene expression data, things like the molecular timetable method and SciY-CLOPS, to analyze these neural activity patterns.
And how accurate was it?
It was highly accurate. They could predict the internal circadian time with a mean absolute error of only about 1.44 hours. It proves that the brain's spatiotemporal activity pattern is so structured and integrated that it functions as its own clock. You can read the time just by looking at the pattern.
Okay, so we need another quick recap. This is huge.
Absolutely. So we learned that functionally similar regions, like those wake-promoting areas, peak together. But opposing functions, like memory encoding versus retrieval or vision versus hearing, they peak in antiphase.
And zooming in, that voxel-wise analysis showed incredible internal detail. We saw peak time gradients within master regions like the SCN and the CA1.
And finally, by normalizing the activity using that cell count ratio, they found they could accurately predict the brain's internal circadian time from just a single snapshot of activity.
So what does this whole brain map of time actually mean for us? Let's talk implications. Medicine has to be a big one.
Oh, the relevance to chronopharmacology giving drugs based on time of day is enormous. We've known for a while that the efficacy and the toxicity of everything from chemotherapy to psychiatric drugs can vary wildly depending on when you take them.
And this atlas basically provides the blueprint for why.
It's the scientific foundation. If you want to target a specific neural population for, say, a psychiatric disorder, you now have a map telling you exactly when that population is at its peak rhythmicity. You can move from a blunt instrument to a chronologically precise one.
It's the foundation for a future of precision medicine. But, you know, as critical thinkers, we have to talk about the blind spots. What are the limitations of the tool they used, of c-Fos?
That's the most important question. And the main issue is that it's an indirect marker, right? So the peak times aren't instantaneous.
There's a lag.
There's a big temporal lag. The c-Fos protein doesn't peak instantly. It takes about 1 to 2 hours after the neuron fires, and then the protein itself has a half-life of another hour and a half or so.
So what we're seeing is a smeared cumulative picture of activity that actually happened a couple hours earlier.
Exactly. It's a temporally lagged and cumulative representation. You can't capture really fast dynamics like what happens during REM sleep or, millisecond level memory signaling.
And I imagine it's also hard to separate the pure circadian drive from just being awake or asleep.
That's the other key point. In constant darkness, activity is super tightly coupled to the sleep-wake state. So these peak times are a combination of both the circadian drive and the behavioral state. It's a map of chronological opportunity, not necessarily the primary driver of that circuit.
That being said, the researchers made this entire incredible data set available to everyone.
They did. They created a fantastic open access database. You can go online, explore the data for any of the 642 regions, and you can even upload your own regions of interest to analyze. It's just an indispensable resource for the whole field.
This whole deep dive really transforms our perspective. I mean, we went from thinking of the brain as having one master clock, the SCN,
to understanding it as a truly integrated system where 79% of its anatomy is operating under this beautiful spatiotemporal coordination.
It's the difference between a single metronome ticking in a room
and a full-blown symphony orchestra. The SCN is the conductor, setting the tempo, but every single instrument, every functional region is playing its own highly specialized, timed, rhythmic part.
Which leads us to our final provocative thought for you, the listener. We saw that clear temporal split, memory encoding in the dentate gyrus peaks mid-subjective day around CT7, but memory retrieval in CA1 peaks late subjective night around CT22.
A huge difference.
So if we can map these peak times in humans, what are the practical implications? Should you be scheduling your most intense study sessions right before your biological night when your brain is primed for encoding? Or maybe should memory rehabilitation therapies be strictly timed to a patient's internal clock to have any real effect?
The future of neuroscience won't just be about asking where something happens in the brain, but with what temporal precision it happens. This atlas is the map that finally makes that question answerable.
Thank you for joining us for this deep dive. We hope you feel a little more informed about the amazing clock inside your own head. We'll see you next time.